Dear colleagues, the new recommendation for performing drug skin tests and the non-irritative concentrations of drugs for skin tests are now published in Allergy. You will find the detailed method for drug skin testing, prick test. For intradermal test, we recommend the ENDA method in injecting a control volume of 0.02 milliliters and detail the method for reading intradermal tests for immediate and delayed hypersensitivities. For patch tests with non-commercial material from a drug product, in case of lyophilizate, you dilute at 10% in petrolatum, while for tablets, you reduce to a fine powder, then dilute at 30% in petrolatum. But it is absolutely mandatory to note the final concentration of the active ingredient. These new guidelines specify the highest non-irritating concentrations for drugs and the specificity of these concentrations according to the number of controls we found in literature. So in 4 different tables, you will find in green recommendations when we found more than 20 controls, in yellow with controls between 10 to 19, and if we found less than 10 controls, the recommendation is in red. And we give non-irritating test concentrations for prick test, patch test, intradermal test with 2 different columns, one for immediate reading and one for delayed readings. On the first table, you will find many concentrations for anti-infectious drugs. On the second one, with miscellaneous drugs as radiocontrast media, corticosteroids, heparins, insulin, cardiovascular drugs. The third one concerns chemotherapies and biologicals. And the last one concerns drugs used perioperatively. The allergy workup follows a stepwise approach. So we provide the workup guided by the initial clinical features and risk stratification. So for each clinical presentation, you will find the mandatory skin test, the recommended dilutions, the reading delay, and their diagnostic value. We also provide the guidance on when and how to complete with adapted drug provocation test. And these detailed recommendations are given for immediate and delayed hypersensitivity. Specifically, we develop recommendations for scars. So we hope that this new standardized method will be helpful for multicenter studies, but also for your daily practice. Thank you for your attention.